we are pleased to present our research dedicated to the role of selectins from platelets in the pathogenesis of systemic lupus. We described a few years ago the role of platelets in the pathogenesis of lupus through an interaction with plasmacytoid dendritic cells. Here, we found a new role of platelets in the pathogenesis of systemic lupus through an interaction with regulatory T cells. Mark is going to present his PhD work from Boston, where he's doing his postdoc.
Thank you, Christophe, for this kind introduction. Hi everyone, my name is Mark Schellinger and I'm pleased to share with you today the result from our latest paper. So in this work, we studied patients with systemic lupus, a potentially life-threatening autoimmune disease. We found that in this patient, their blood platelets were activated. Upon activation, these platelets express P-selectin, a protein that gives them the ability to specifically interact with some immune cell. More specifically, we found that blood platelets in patients with lupus interacted with T regulatory cells. T regulatory cells are very important cells which are responsible for the control of the immune response, which is known to be dysregulated with patients with systemic lupus. We found that the interaction between the blood platelets and the T regulatory cells induced an intracellular signaling that ultimately led to the dysfunction of the T regulatory cell and its inability to suppress the immune response. We found that this phenomenon was particularly increased in patients with high disease activity. Finally, we studied a mouse model of systemic lupus. We found that these mice had, similarly to patients, increased level of P-selectin. Interestingly, when we treated the mice with an anti-P-selectin antibody, we were able to rescue the Treg function and to restore immune regulation. Indeed, the mice treated with the anti-P-selectin antibody showed improvement in cardinal feature of the disease. Globally, our results suggest that P-selectin blockade has a high potential in the treatment of systemic lupus and potentially other autoimmune diseases. These results are exciting on several aspects. First, we found that blocking P-selectin is able to restore immune regulation and improve disease activity. Second, P-selectin has been demonstrated to be a key factor in the progression of atherosclerosis and heart disease in patients with systemic lupus. Therefore, P-selectin blockade is likely to be a multimodal therapy that would improve the immune regulation and the disease activity, but also decrease the risk of heart disease in these patients. And finally, I would like to thank all the people who made this study possible, our financial sponsors, as well as, of course, the patients.